Rahim. I welcome you in the third International Conference on Applied Zoology 2020, organized by the Applied Zoological Society of Pakistan and Qaid-e-Azam University, Islamabad, Pakistan. I am Dr. Nosheen Aslam from Department of Biochemistry, Government College University, Faisalabad, Pakistan, and my topic of presentation is anti-adhesion and antibiofilm potential of Fagonia indica from Cholistan desert against clinical multidrug resistant bacteria. Pakistan is one of the richest countries in medicinal flora. Cholistan desert covered the 2.6 million hectares of Pakistan. Fagonia indica present in subtropical and hot temperature regions like Morocco, Tunisia, Algeria, Cyprus, Saudi Arabia, and dry regions of salty rocks of India and western Pakistan. It is mostly used in the Indo-Pak for the treatment of fever, urinary discharges, asthma, liver troubles, tumors, and peptic ulcer diseases. Antimicrobial activity of Fagonia indica has been reported against different microbes like Salmonella typhi, Escherichia coli, and Staphylococcus epidermidis. So, keeping in view the threat of multi-drug resistant bacteria, present study has been designed to investigate the in vitro antibacterial activity of different extracts of Fagonia indica prepared by different extraction methods against multi-drug resistant bacteria. Effect of Fagonia indica on biofilm formation and adhesion was also evaluated in our study. First of all, Fagonia indica plant was collected randomly from Tehsil Yazman, Cholistan district, Bahawalpur, Punjab, Pakistan, and after the identification, the collected plants were rinsed with distilled water and finally dried in shade. Dried materials, that is the roots and aerial parts, were ground in a mixture grinder and into fine powder. Powder material of each plant part was sequentially extracted by softlet apparatus using different solvents in increasing polarity. After plant collection, the antibacterial activity of plant extract was evaluated against 4 multidrug resistant clinical isolates, that is Staphylococcus aureus, Pseudomonas aeruginosa, Escherichia coli, and Klebsiella pneumonia. After this inoculum preparation, each bacterial strain was subcultured in LB broth at 37 degrees centigrade. The growth of bacteria was harvested. Antibacterial activity of different plant extracts was determined using the agar-agar diffusion method. Next, the minimum inhibitory concentration and minimum bacterial concentration of plant extract was determined by broth microdilution method. DMSO was used as a negative control and ampicillin was used as positive control. Detection of cellular protein leakage. In order to detect the disintegration of cell envelope after the treatment of cells with plant extract exhibiting the lowest minimum inhibitory cellular protein leakage was measured. The absorbance of the suspensions was measured at 595 nanometer. Then the cell attachment assay was used to determine the effect of plant extract on adhesion of bacteria to polystyrene surfaces. Negative and positive control were also included in each experiment. Biofilm formation was used to assess the effect of plant extracts on biofilm formation of target bacteria. Finally, the absorbance of the eluted suspensions was measured at 546 nanometer. All the experiments were performed in triplicate. Now, the highest yield was found for chloroform extract of Fagonia made from roots, followed by methanolic extract obtained from aerial parts. Our results revealed that Fagonia extract has exhibited antibacterial activity in a dose-dependent manner, since increasing the concentration of extract increase the diameter zone of inhibition. Diameter of zone of inhibition range from 5 to 15 millimeter, and the largest zone of inhibition was observed against Staphylococcus aureus using chloroform extract of plant made from aerial parts. The results also indicate that extract made from aerial parts of Fagonia indica were more effective against tested bacteria as compared to the extract made from roots. Next, the results indicated that minimum inhibitory concentration values range between 125 to 1000 microgram per ml, and the least values were observed for chloroform extract made from aerial parts against Staphylococcus aureus and Pseudomonas aeruginosa. Our data displayed that minimum bacterial concentration values range between 500 to 4000 microgram per ml, and the presented value was 2 to 8 times greater than their respective MIC value, as you can see in the table 2. The results have indicated that these bacterial cells treated with chloroform extract of Fagonia resulted in significant increase in protein leakage as compared to control. It was observed that maximum protein leakage was observed for the Klebsiella pneumonia, followed by Staphylococcus aureus as displayed in figure 1. The results revealed that after 12 hours of incubation in the presence of chloroform extract, bacterial adhesion to the plastic surface was significantly reduced when compared with control cells. This inhibition of adhesion was found to be 43.5 to 53.5%. Static biofilm formation assay was employed to quantify biofilm formation in the presence and absence of sub-inhibitory concentration of chloroform extract of Fagonia. This data also indicated that biofilm forming capacity was increased in a time-dependent manner among multidrug resistant bacteria, and maximum capacity was observed for Pseudomonas aeruginosa, followed by Klebsiella pneumonia. It was also observed that sub-inhibitory concentration of chloroform extract of Fagonia significantly reduced the biofilm forming capacity of tested bacteria, and percentage inhibition of biofilm formation was found to be 30.33 to 71.84% as compared with control cells. Our data also revealed that after 48 hours of incubation, greater inhibition was observed for Staphylococcus aureus, that is 71.84%, followed by Pseudomonas pneumonia. Our study clearly suggested that efficient role of Fagonia indica extract as antiadhesion and antibiofilm agents against multidrug resistant bacteria. Biofilm formation is considered to be a major public health concern, since more than 80% of microbial infections of human beings involve biofilm formers. Therefore, current study is significant, mentioning the role of Fagonia collected from Cholistan desert biofilm inhibitors. However, in future there is need to identify individual compounds from this plant responsible for this inhibition, and also their molecular mechanism of action against antibiotic resistant bacteria. Thank you.